Hi everyone and welcome back to STEM Revise. Today we're going to be talking about DNA replication as part of our A-level biology series. So DNA replication takes place through a process called semi-conservative replication. And for this to take place, there are three main requirements. The first of which is the presence of four types of nucleotide: adenine, thymine, cytosine, and guanine. These all must be available. These nucleotides are the building blocks that form the new DNA strands. Each nucleotide pairs with its complementary base on the original DNA strand, A with T and C with G. Secondly, the enzyme DNA polymerase is essential for replication. This enzyme binds to the DNA strand and helps add nucleotides to form the new complementary strand. DNA polymerase also has a proofreading function, which corrects errors to ensure the DNA copy is as accurate as possible. Finally, a source of chemical energy is needed to power the entire process. This energy typically comes from ATP and is used to form the phosphodiester bonds between nucleotides, allowing the DNA strands to elongate. These three components, nucleotides, DNA polymerase, and chemical energy, are all crucial for the accurate replication of DNA. Let's actually discuss the method by which DNA replicates. First up, DNA helicase is the enzyme that starts the process. It does this by breaking the hydrogen bonds between complementary base pairs, adenine with thymine and cytosine with guanine. This action separates the two DNA strands, preparing them to act as templates for the new DNA. As the hydrogen bonds break, the double helix unwinds into its two separate strands. Imagine the DNA molecule as a zipper. DNA helicase essentially unzips it, exposing the nitrogenous bases on each strand. Each strand now has the potential to guide the formation of a new complementary strand. With the DNA strands separated, each one acts as a template. Three nucleotides in the nucleus begin to bind to their complementary bases on each exposed strand. This binding follows specific base pairing rules, as we discussed previously. This precise matching is crucial for creating an exact copy of the original DNA sequence. Next, DNA polymerase, an essential enzyme, facilitates the joining of nucleotides through a condensation reaction. This reaction forms strong covalent bonds, called phosphodiester bonds, between the sugar and phosphate groups of each nucleotide. These bonds create the sugar-phosphate backbone of the new strand, stabilizing the DNA structure. DNA polymerase also has a proofreading role, as we discussed before. Finally, we reach the result of DNA replication. Each new DNA molecule contains one original strand and one newly synthesized strand. This is known as semi-conservative replication, meaning half the original DNA is conserved in each new molecule. This method ensures genetic continuity, passing down identical DNA from cell to cell and from generation to generation. So in order to actually provide some evidence for semi-conservative replication, scientists conducted an experiment where they grew bacteria in two different types of nitrogen. Some bacteria were grown in a medium containing a lighter nitrogen isotope, 14N, while others were grown in a medium containing a heavier nitrogen isotope, 15N. This meant that all the DNA in each group would contain either 14N or 15N, making them distinguishable by their density. Next, the bacteria with 15N in their DNA were transferred to a new medium with only 14N available. They were left to reproduce so that any new DNA strands would incorporate the lighter 14N, while the original strands still contained 15N. After a round of DNA replication, the DNA was extracted from the bacteria and centrifuged. In the centrifuge, DNA with both 15N and 14N settled in a band between pure 15N and pure 14N. This intermediate position indicated that each new DNA molecule contained one original 15N strand and one new 14N strand. This result supported the semi-conservative replication model, meaning that each new DNA molecule consists of one old strand and one new strand. The experiment showed that DNA doesn't copy itself entirely new or mix randomly, but conserves half of the original strand in each new DNA molecule that's produced. So, thank you very much for taking the time to watch this video on DNA replication. Make sure to like, comment and subscribe, and also feel free to check out any other videos on this channel.